Assalamu alaikum. Important questions of microbiology 2022. No more questions required. Just study this with answers. See, my dear brothers and sisters, for your easy convenience, for your easiness to understand the concept very clear, crystal clear, and maybe day after tomorrow is our examination of microbiology. So, for your happiness, by taking your side, I have printed some of the questions in microbiology with answers. So practice these questions without fail with 2 marks. Definitely you will pass the examination. Okay. Yeah. The first question goes to what is pure culture? Enlist method for isolation of pure culture. Then describe any two industrial important techniques preserving bacteria. See here, first you have to write here what is this pure culture. Okay, means we are developing only single type of individual there. Okay, single type of cell, single type of bacteria, and you have to list the isolation of a pure culture like streak plate method. Okay, then spreader method. Some methods are there. First you write all those method, then you explain any two. You don't worry, just after these 5 questions I will go to the PDF of that, means the question paper which I had the answers for these questions. Next to these 5 questions there will be answers for these 5 questions. The answers may be jumbled, means there will be not in ordered way. First question, second question may be mismatched, but you have to find that. Okay, that's very easy. The next question is in that you have to write the industrial important techniques preserving bacteria. Next, explain the principal procedural applications of sterilization using hot air one. Sometimes they will ask in the hot air one or autoclave, or they will ask steam distillation method. Okay, write a procedure, principle, and application of thermal distillation. Sorry, thermal sterilization method. Give principle, construction, working of a thermal method of sterilization. There you have to write hot air one. It is understandable question. Then explain different methods of evaluation of disinfectants. Phenol coefficient is compulsory here. Then various methods involved in identification of the bacteria. Many methods are there. How can you identify the bacteria? You have to write those all methods of identification of the bacteria. First you have to write what is this bacteria, then classify the bacteria, then you go for identification. Then what is virus? Morphology and replication of virus. You have to tell what is this virus, obligate parasite you have to write, and some morphology of that virus you have to write. Then you have to write the structure, beautiful structure of the naked virus. Clear? Naked virus or packed virus, means a virus which has a capsomers. Both you can, anyone you can write. If they ask compulsory naked, means you have to write compulsory naked, but most of the questions they have asked only viruses. Then you have to draw a diagram of that, explain the parts, go for morphology, replication. Then you have to, one more question you have to understand here. If they not ask replication, means cultivation of virus is fixed here. Now see, I will tell you some answers for this. Let's know that answers. Yes, microbiology 10 marks answer. See, this is the answer for first question. Isolation and preservation of a pure culture. Just you can take a screenshot of this. This is streak plate method and principle for streak plate method, then pour plate method. You have to write this one all. Okay, better what I will do means I will add this in the description. Go and download this. Okay. These all are 10 marks. Some questions are selected. All questions are for 10 marks, and I will add this in the description. Evaluation of antimicrobial agent. It is very important one. And phenol coefficient method. You have to write this compulsory. Advantages of phenol coefficient test. Then staining techniques, means this staining techniques, it is answer for the identification of the bacteria. Gram stain, first you have to write a simple staining, then complex staining. In that complex staining, gram staining of bacteria, positive and negative. In big test for 10 marks you have to write this all. Then this is the virus structure. This is the enveloped virus. So go through this. Clear? Now in next slide I will show you 5 marks answers. Yes, now these are the 5 marks, very important. Explain difference between gram positive and gram negative cell wall. See, in the year 2021 this gram positive and gram negative was asked in 10 marks. You don't worry, your 5 marks questions, sometimes what they will do, they will jump to the 5 marks, they will jump to the 10 marks, sometimes in 10 marks they will jump to 5 marks. Okay, this alternating pathway or alternation passion will be observed in your question paper every year, but without these questions there will be no chance of getting other questions. Okay, today's question paper, that is of PP physical pharmaceutics 1, no, it's a little bit difficult paper. Why? Means all the questions are newly prescribed one, discussed one. Okay, so in the history from year 2015 to till 2022, that is the toughest paper in PP. Okay, but you can pass that if you study little forwarded, means you can easily pass that subject. Now leave that PP, come here for microbiology. Definitely these questions will be asked, you don't worry, forget that one question paper they will be framed that way, means after 10 years that question paper has come in this passion. Okay, so leave that. Next question, difference between prokaryotes and eukaryotes, very important one. Then you have to write a diagram for the prokaryote, then you have to go for eukaryotes, there you have to write a diagram, is compulsory, some write morphology of that prokaryotes. Okay, and eukaryotes, these are very important. Then at last you make 2 columns, differences columns, you just explain in short what you had wrote there. Then explain principle and procedure involved in MRVP test, very important. Then discuss sterilization by radiation. Then basic requirements to establish cell culture laboratory. Then write the principal procedure of acid fast staining, gram staining, these are very important. Clear? Next, yes, now I will give you answer for these. See here, these are the answers. Yes, now 7th question, describe bacterial growth curve. You have to write what is this bacterial growth curve. 4 phases are there, write these phases with a graph, explain those phases. Next one, principal procedure, merits and demerits of membrane filtration, very important one, this is membrane filtration. The 9th one, explain principle involved in microbial assay of a streptomycin. Sometimes they will ask directly streptomycin, sometimes what they will do, they will ask antibiotic. See, both, for both these are the same answer, any antibiotic. You take amoxicillin, streptomycin, or cephaloxime, cephalosporin, everything, for all the microbial assay there is the same one, antibiotic. But if they ask for vitamins means vitamin B6, B12, any type of the vitamin, okay, you have to write the vitamin microbial assay. Next, the main sources of contamination of the aseptic room. What are the main sources are there? Okay, industrial sources are there, then human source are there, you have to write these all. Then how can you prevent that, that is also very important here. Then outline working of TEM and SEM microscopy, very important one. Then define and classify culture media with example. Yeah, write a note on cultivation of virus. I have told you, if they are not asking 10 marks, means definitely they will ask here. Okay, maybe there is a chance of asking both the questions, one is replication and cultivation, so be perfect in both. Next one, explain principle involved in the assay of vitamin B12, that is of vitamins. Then explain the principle involved in autoclaving. Yes. Then explain the different sources and types of microbial contamination of pharmaceutical preparations or pharmaceutical aids. Okay. Then write briefly general procedure for cell culture. General procedure for cell culture. How your cell culture should be. How can you develop that cell culture? Then discuss the different methods of microbial assay of antibiotic. See, antibiotics sometimes they ask in this fashion, antibiotic. Sometimes what they will do, they will ask directly streptomycin. Then principle of sterility testing. Sterility testing. It is also very important. Design and working of laminar airflow. Laminar airflow, or they will ask in this way. Explain design and working of aseptic equipment. Next, factors affecting gaseous sterilization. In the year 2021, this question has asked, okay, 2021, what's a gaseous sterilization? Mention ideal characteristics of disinfectant. However, disinfectant should be. Don't go directly for the characteristic. First you write disinfectant, then you write some of the basic functions of that disinfectant, then you go for ideal characteristics. Or you can write first definition of the disinfectant, then you go for characteristics, explain those characteristics, then go for functions of that disinfectant. Then explain the cultivation of anaerobic bacteria. Glass pack method is there, no, or glass jar method. You have to explain this glass jar method. Principle involved in the sterilization by filtration. It is also very important. One is filtration, another one is radiation. From these two, one is fixed for your examination. Yes, these are the 10 mark and 5 marks very important questions. Let's see in the next PDF the answer for these 4 questions. See, one more note I want to tell you, you should read every question of this PDF. No more skipping here. And 2 mark questions, it's very important to pass to score 50 plus. 2 marks questions, these are the pillars for your 10 marks and 5 marks. Okay, if you remove those pillars means there will be no proper standing position. So please, my request to you all to practice these 2 marks without skipping. Already I made a PDF for that. PDF way I have uploaded the video on my channel Let's Join Pharmacy, on my YouTube channel Let's Join Pharmacy. You can just go and download, see that, observe that. You can take screenshot, it's better for you, or simply I will add in the description, go and download that. Now let's see the answers for these 4 questions. See, these are 5 marks. Difference between gram positive and gram negative cell wall. You have to write until here. Then difference between prokaryotes and eukaryotes. See, make 2 columns compulsory at last. Then write a principle and procedure of radiation. Then laminar airflow. General requirements. What general requirements we have to require? No, that is here, this answer. You have to write dissolve. General requirements of cell culture laboratory. This one, disinfectant, Bunsen time. This is acid fast staining. This bacterial growth curve. Then methods for sterility testing, membrane filtration method, this one. This different methods of microbial assay of antibiotic, this one. Electron microscopy, SEM, TEM. Okay, just to practice this, I will add this one in the description. And this one is anaerobic jar method or gas pack method. This one is filtration. Everything I have made a PDF form and uploaded. This is laminar airflow. Factors affecting gaseous sterilization, very important one. Some 2 or 3 question answers I have not printed here, I have not given, but you can ask me privately, inshallah I will find and give you. Thank you. Assalamu alaikum.